Nicotinamide adenine dinucleotide is a coenzyme found in all living cells. The compound is a dinucleotide, since it consists of two nucleotides joined through their phosphate groups. One nucleotide contains an adenine base and the other nicotinamide. Nicotinamide adenine dinucleotide exists in two forms, an oxidized and reduced form abbreviated as NAD+ and NADH respectively. In metabolism, nicotinamide adenine dinucleotide is involved in redox reactions, carrying electrons from one reaction to another. The coenzyme is, therefore, found in two forms in cells. NAD+ is an oxidizing agent. It accepts electrons from other molecules and becomes reduced. This reaction forms NADH, which can then be used as a reducing agent to donate electrons. These electron transfer reactions are the main function of NAD. However, it is also used in other cellular processes, the most notable one being a substrate of enzymes that add or remove chemical groups from proteins, in post-translational modifications. Because of the importance of these functions, the enzymes involved in NAD metabolism are targets for drug discovery. In organisms, NAD can be synthesized from simple building blocks from the amino acids tryptophan or aspartic acid. In an alternative fashion, more complex components of the coenzymes are taken up from food as the vitamin called niacin. Similar compounds are released by reactions that break down the structure of NAD. These preformed components then pass through a salvage pathway that recycles them back into the active form. Some NAD is also converted into nicotinamide adenine dinucleotide phosphate. The chemistry of this related coenzyme is similar to that of NAD, but it has different roles in metabolism. Although NAD+ is written with a superscript plus sign because of the formal charge on a particular nitrogen atom, at physiological pH for the most part it is actually a singly charged anion, while NADH is a doubly charged anion. NAD is considered the V factor required for the growth of Haemophilus influenzae. Physical and chemical properties. Nicotinamide adenine dinucleotide, like all dinucleotides, consists of two nucleotides joined by a pair of bridging phosphate groups. The nucleotides consist of ribose rings, one with adenine attached to the first carbon atom and the other with nicotinamide at this position. The nicotinamide moiety can be attached in two orientations to this anomeric carbon atom. Because of these two possible structures, the compound exists as two diastereomers. It is the I-squared nicotinamide diastereomer of NAD+ that is found in organisms. These nucleotides are joined together by a bridge of two phosphate groups through the 5' carbons. In metabolism, the compound accepts or donates electrons in redox reactions. Such reactions involve the removal of two hydrogen atoms from the reactant, in the form of a hydride ion, and a proton. The proton is released into solution, while the reductant RH2 is oxidized and NAD+ reduced to NADH by transfer of the hydride to the nicotinamide ring. RH2 plus NAD plus NADH plus H plus R. From the hydride electron pair, one electron is transferred to the positively charged nitrogen of the nicotinamide ring of NAD+, and the second hydrogen atom transferred to the C4 carbon atom opposite this nitrogen. The midpoint potential of the NAD+/NADH redox pair is a 0.32 volts, which makes NADH a strong reducing agent. The reaction is easily reversible when NADH reduces another molecule and is reoxidized to NAD+. This means the coenzyme can continuously cycle between the NAD+ and NADH forms without being consumed. In appearance, all forms of this coenzyme are white amorphous powders that are hygroscopic and highly water-soluble. The solids are stable if stored dry and in the dark. Solutions of NAD+ are colorless and stable for about a week at 4 degrees Celsius and neutral pH, but decompose rapidly in acids or alkalis. Upon decomposition, they form products that are enzyme inhibitors. Both NAD+ and NADH strongly absorb ultraviolet light because of the adenine. For example, peak absorption of NAD+ is at a wavelength of 259 nanometers, with an extinction coefficient of 16,900 mR1 kmR1. NADH also absorbs at higher wavelengths, with a second peak in UV absorption at 339 nm with an extinction coefficient of 6220 mR1 kmR1. This difference in the ultraviolet absorption spectra between the oxidized and reduced forms of the coenzymes at higher wavelengths makes it simple to measure the conversion of one to another in enzyme assays by measuring the amount of UV absorption at 340 nm using a spectrophotometer. NAD+ and NADH also differ in their fluorescence. NADH in solution has an emission peak at 460 nm and a fluorescence lifetime of 0.4 nanoseconds, while the oxidized form of the coenzyme does not fluoresce. The properties of the fluorescence signal changes when NADH binds to proteins, so these changes can be used to measure dissociation constants, which are useful in the study of enzyme kinetics. These changes in fluorescence are also used to measure changes in the redox state of living cells, through fluorescence microscopy. Concentration and state in cells. In rat liver, the total amount of NAD+ and NADH is approximately 1 AI 1/4 mol per gram of wet weight, about 10 times the concentration of NADP+ and NADPH in the same cells. The actual concentration of NAD+ in cell cytosol is harder to measure, with recent estimates in animal cells, ranging around 0.3 ohm, and approximately 1.0 to 2.0 ohm in yeast. However, more than 80% of NADH fluorescence in mitochondria is from bound form, so the concentration in solution is much lower. Data for other compartments in the cell are limited, although, in the mitochondrion the concentration of NAD+ is similar to that in the cytosol. This NAD+ is carried into the mitochondrion by a specific membrane transport protein, since the coenzyme cannot diffuse across membranes. The balance between the oxidized and reduced forms of nicotinamide adenine dinucleotide is called the NAD+/NADH ratio. This ratio is an important component of what is called the redox state of a cell, a measurement that reflects both the metabolic activities and the health of cells. The effects of the NAD+/NADH ratio are complex, controlling the activity of several key enzymes, including glyceraldehyde 3-phosphate dehydrogenase and pyruvate dehydrogenase. In healthy mammalian tissues, estimates of the ratio between free NAD+ and NADH in the cytoplasm typically lie around 700. The ratio is thus favorable for oxidative reactions. The ratio of total NAD+/NADH is much lower, with estimates ranging from 3/10 in mammals. In contrast, the NADP+/NADPH ratio is normally about 0.005, so NADPH is the dominant form of this coenzyme. These different ratios are key to the different metabolic roles of NADH and NADPH. Biosynthesis, NAD+ is synthesized through two metabolic pathways. It is produced either in a de novo pathway from amino acids or in salvage pathways by recycling preformed components such as nicotinamide back to NAD+. De novo production. Most organisms synthesize NAD+ from simple components. The specific set of reactions differs among organisms, but a common feature is the generation of quinolinic acid from an amino acid either tryptophan in animals and some bacteria, or aspartic acid in some bacteria and plants. The quinolinic acid is converted to nicotinic acid mononucleotide by transfer of a phosphoribose moiety. An adenylate moiety is then transferred to form nicotinic acid adenine dinucleotide. Finally, the nicotinic acid moiety in NAD is amidated to a nicotinamide moiety, forming nicotinamide adenine dinucleotide. In a further step, some NAD+ is converted into NADP+ by NAD+ kinase, which phosphorylates NAD+. In most organisms, this enzyme uses ATP as the source of the phosphate group, although several bacteria such as Mycobacterium tuberculosis and a hyperthermophilic archaeon Pyrococcus horaceae, use inorganic polyphosphate as an alternative phosphoryl donor. Salvage pathways. Besides assembling NAD+ de novo from simple amino acid precursors, cells also salvage preformed compounds containing nicotinamide. Although other precursors are known, the three natural compounds containing the nicotinamide ring and used in these salvage metabolic pathways are nicotinic acid, nicotinamide and nicotinamide riboside. These compounds can be taken up from the diet, where the mixture of nicotinic acid and nicotinamide are called vitamin B3 or nicene. However, these compounds are also produced within cells, when the nicotinamide moiety is released from NAD plus in ADP ribose transfer reactions. Indeed, the enzymes involved in these salvage pathways appear to be concentrated in the cell nucleus, which may compensate for the high level of reactions that consume NAD plus in this organelle. Cells can also take up extracellular NAD plus from their surroundings. Despite the presence of the de novo pathway, the salvage reactions are essential in humans. A lack of nicene in the diet causes the vitamin deficiency disease pellagra. This high requirement for NAD+ results from the constant consumption of the coenzyme in reactions such as post-translational modifications, since the cycling of NAD+ between oxidized and reduced forms in redox reactions does not change the overall levels of the coenzyme. The salvage pathways used in microorganisms differ from those of mammals. Some pathogens, such as the yeast Candida glabrata and the bacterium Haemophilus influenzae are NAD plus autotroph they cannot synthesize NAD plus AA but possess salvage pathways and thus are dependent on external sources of NAD plus or its precursors. Even more surprising is the intracellular pathogen Chlamydia trachomatis, which lacks recognizable candidates for any genes involved in the biosynthesis or salvage of both NAD plus and NADP plus, and must acquire these coenzymes from its host. Functions. Nicotinamide adenine dinucleotide has several essential roles in metabolism. It acts as a coenzyme in redox reactions, as a donor of ADP ribose moieties in ADP ribosylation reactions, as a precursor of the second messenger molecule cyclic ADP ribose, as well as acting as a substrate for bacterial DNA ligases and a group of enzymes called sirtuins that use NAD+ to remove acetyl groups from proteins. In addition to these metabolic functions, NAD+ emerges as an adenine nucleotide that can be released from cells spontaneously and by regulated mechanisms, and can therefore have important extracellular roles. Oxidoreductase binding of NAD. The main role of NAD+ in metabolism is the transfer of electrons from one molecule to another. Reactions of this type are catalyzed by a large group of enzymes called oxidoreductases. The correct names for these enzymes contain the names of both their substrates, for example NADH ubiquinone oxidoreductase catalyzes the oxidation of NADH by coenzyme Q. However, these enzymes are also referred to as dehydrogenases or reductases, with NADH ubiquinone oxidoreductase commonly being called NADH hydrogenase or sometimes coenzyme Q reductase. When bound to a protein, NAD+ and NADH are usually held within a structural motif known as the Rossmann fold. The motif is named after Michael Rossmann, who was the first scientist to notice how common this structure is within nucleotide-binding proteins. This fold contains 3 or more parallel beta strands linked by 2 alpha helices in the order beta-alpha-beta-alpha-beta. This forms a beta sheet flanked by a layer of alpha helices on each side. Because each Rossmann fold binds one nucleotide, binding domains for the dinucleotide NAD+ consist of 2 paired Rossmann folds, with each fold binding one nucleotide within the cofactor. However, this fold is not universal among NAD-dependent enzymes, since a class of bacterial enzymes involved in amino acid metabolism have recently been discovered that bind the coenzyme, but lack this motif. When bound in the active site of an oxidoreductase, the nicotinamide ring of the coenzyme is positioned so that it can accept a hydride from the other substrate. Since the C4 carbon that accepts the hydrogen is prochiral, this can be exploited in enzyme kinetics to give information about the enzyme's mechanism. This is done by mixing an enzyme with a substrate that has deuterium atoms substituted for the hydrogens, so the enzyme will reduce NAD+ by transferring deuterium rather than hydrogen. In this case, an enzyme can produce one of 2 stereoisomers of NADH. In some enzymes the hydrogen is transferred from above the plane of the nicotinamide ring. These are called class A oxidoreductases, whereas class B enzymes transfer the atom from below. Despite the similarity in how proteins bind the two coenzymes, enzymes almost always show a high level of specificity for either NAD+ or NADP+. This specificity reflects the distinct metabolic roles of the respective coenzymes, and is the result of distinct sets of amino acid residues in the two types of coenzyme binding pocket. For instance, in the active site of NADP-dependent enzymes, an ionic bond is formed between a basic amino acid side chain and the acidic phosphate group of NADP+. On the converse, in NAD-dependent enzymes the charge in this pocket is reversed, preventing NADP+ from binding. However, there are a few exceptions to this general rule, and enzymes such as aldose reductase, glucose 6-phosphate dehydrogenase, and methyl N-tetrahydrofolate reductase can use both coenzymes in some species. Role in redox metabolism. The redox reactions catalyzed by oxidoreductases are vital in all parts of metabolism, but one particularly important area where these reactions occur is in the release of energy from nutrients. Here, reduced compounds such as glucose and fatty acids are oxidized, thereby releasing energy. This energy is transferred to NAD+ by reduction to NADH, as part of beta-oxidation, glycolysis, and the citric acid cycle. In eukaryotes, the electrons carried by the NADH that is produced in the cytoplasm are transferred into the mitochondrion by mitochondrial shuttles, such as the malate aspartate shuttle. The mitochondrial NADH is then oxidized in turn by the electron transport chain, which pumps protons across a membrane and generates ATP through oxidative phosphorylation. These shuttle systems also have the same transport function in chloroplasts. Since both the oxidized and reduced forms of nicotinamide adenine dinucleotide are used in these linked sets of reactions, the cell maintains significant concentrations of both NAD+ and NADH, with the high NAD+/NADH ratio allowing this coenzyme to act as both an oxidizing and a reducing agent. In contrast, the main function of NADPH is as a reducing agent in anabolism, with this coenzyme being involved in pathways such as fatty acid synthesis and photosynthesis. Since NADPH is needed to drive redox reactions as a strong reducing agent, the NADP+/NADPH ratio is kept very low. Although it is important in catabolism, NADH is also used in anabolic reactions, such as gluconeogenesis. This need for NADH in anabolism poses a problem for prokaryotes growing on nutrients that release only a small amount of energy. For example, nitrifying bacteria such as Nitrobacter oxidize nitrite to nitrate, which releases sufficient energy to pump protons and generate ATP, but not enough to produce NADH directly. As NADH is still needed for anabolic reactions, these bacteria use a nitrite oxidoreductase to produce enough proton motive force to run part of the electron transport chain in reverse, generating NADH. Non-redox roles. The coenzyme NAD+ is also consumed in ADP ribose transfer reactions. For example, enzymes called ADP ribosyl transferases add the ADP ribose moiety of this molecule to proteins, in a post-translational modification called ADP ribosylation. ADP ribosylation involves either the addition of a single ADP ribose moiety, in mono-ADP ribosylation, or the transferal of ADP ribose to proteins in long-branched chains, which is called poly-ADP ribosylation. Mono-ADP ribosylation was first identified as the mechanism of a group of bacterial toxins, notably cholera toxin, but it is also involved in normal cell signaling. Poly-ADP ribosylation is carried out by the poly-ADP ribose polymerases. The poly-ADP ribose structure is involved in the regulation of several cellular events and is most important in the cell nucleus, in processes such as DNA repair and telomere maintenance. In addition to these functions within the cell, a group of extracellular ADP ribosyl transferases has recently been discovered, but their functions remain obscure. NAD+ may also be added on to cellular RNA as a 5-terminal modification. Another function of this coenzyme in cell signaling is as a precursor of cyclic ADP ribose, which is produced from NAD+ by ADP ribosyl cyclases, as part of a second messenger system. This molecule acts in calcium signaling by releasing calcium from intracellular stores. It does this by binding to and opening a class of calcium channels called ryanodine receptors, which are located in the membranes of organelles, such as the endoplasmic reticulum. NAD+ is also consumed by sirtuins, which are NAD-dependent deacetylases, such as SIR2. These enzymes act by transferring an acetyl group from the substrate protein to the ADP ribose moiety of NAD+. This cleaves the coenzyme and releases nicotinamide and O-acetyl ADP ribose. The sirtuins mainly seem to be involved in regulating transcription through deacetylating histones and altering nucleosome structure. However, non-histone proteins can be deacetylated by sirtuins as well. These activities of sirtuins are particularly interesting because of their importance in the regulation of aging. Other NAD-dependent enzymes include bacterial DNA ligases, which join two DNA ends by using NAD+ as a substrate to donate an adenosine monophosphate moiety to the 5' phosphate of one DNA end. This intermediate is then attacked by the 3' hydroxyl group of the other DNA end, forming a new phosphodiester bond. This contrasts with eukaryotic DNA ligases, which use ATP to form the DNA-AMP intermediate. Extracellular actions of NAD+. In recent years, NAD+ has also been recognized as an extracellular signaling molecule involved in cell-to-cell communication. NAD+ is released from neurons in blood vessels, urinary bladder, large intestine, from nersecretory cells, and from brain synaptosomes, and is proposed to be a novel neurotransmitter that transmits information from nerves to effector cells in smooth muscle organs. Further studies are needed to determine the underlying mechanisms of its extracellular actions and their importance for human health and diseases. Pharmacology and medical uses. The enzymes that make and use NAD+ and NADH are important in both pharmacology and the research into future treatments for disease. Drug design and drug development exploits NAD+ in three ways: as a direct target of drugs, by designing enzyme inhibitors or activators based on its structure that change the activity of NAD-dependent enzymes, and by trying to inhibit NAD+ biosyntheses. The coenzyme NAD+ is not itself currently used as a treatment for any disease. However, it is being studied for its potential use in the therapy of neurodegenerative diseases such as Alzheimer's and Parkinson's disease. Evidence on the benefit of NAD+ in neurodegeneration is mixed. Some studies in mice have produced promising results, whereas a placebo-controlled clinical trial in humans failed to show any effect. NAD+ is also a direct target of the drug isoniazid, which is used in the treatment of tuberculosis, an infection caused by Mycobacterium tuberculosis. Isoniazid is a prodrug, and once it has entered the bacteria, it is activated by a peroxidase enzyme, which oxidizes the compound into a free radical form. This radical then reacts with NADH, to produce adducts that are very potent inhibitors of the enzymes enoylasyl carrier protein reductase, and dihydrofolate reductase. In one experiment, mice given NAD for 1 week had improved nuclear mitochondrial communication. Since a large number of oxidoreductases use NAD+ and NADH as substrates, and bind them using a highly conserved structural motif, the idea that inhibitors based on NAD+ could be specific to one enzyme is surprising. However, this can be possible. For example, inhibitors based on the compounds mycophenolic acid and thiazuvurin inhibit IMP dehydrogenase at the NAD+ binding site. Because of the importance of this enzyme in purine metabolism, these compounds may be useful as anti-cancer, antiviral, or immunosuppressive drugs. Other drugs are not enzyme inhibitors, but instead activate enzymes involved in NAD+ metabolism. Sirtuins are a particularly interesting target for such drugs, since activation of these NAD-dependent deacetylases extends lifespan. Compounds such as resveratrol increase the activity of these enzymes, which may be important in their ability to delay aging in both vertebrate and invertebrate model organisms. Because of the differences in the metabolic pathways of NAD+ biosynthesis between organisms, such as between bacteria and humans, this area of metabolism is a promising area for the development of new antibiotics. For example, the enzyme nicotinamide, which converts nicotinamide to nicotinic acid, is a target for drug design, as this enzyme is absent in humans but present in yeast and bacteria. History. The coenzyme NAD+ was first discovered by the British biochemists Arthur Hardin and William John Young in 1906. They noticed that adding boiled and filtered yeast extract greatly accelerated alcoholic fermentation in unboiled yeast extracts. They called the unidentified factor responsible for this effect a coferment. Through a long and difficult purification from yeast extracts, this heat-stable factor was identified as a nucleotide sugar phosphate by Hans von Euler-Chelpin. In 1936, the German scientist Otto Heinrich Warburg showed the function of the nucleotide coenzyme in hydride transfer and identified the nicotinamide portion as the site of redox reactions. A source of nicotinamide was identified in 1938, when Konrad Elvigem purified niacin from liver and showed this vitamin contained nicotinic acid and nicotinamide. Then, in 1939, he provided the first strong evidence that niacin was used to synthesize NAD+. In the early 1940s, Arthur Kornberg made another important contribution towards understanding NAD+ metabolism, by being the first to detect an enzyme in the biosynthetic pathway. Subsequently, in 1949, the American biochemists Morris Friedkin and Albert L. Leninger proved that NADH-linked metabolic pathways such as the citric acid cycle with the synthesis of ATP in oxidative phosphorylation. Finally, in 1959, Jack Prius and Philip Handler discovered the intermediates and enzymes involved in the biosynthesis of NAD+. Consequently, de novo synthesis is often called the Prius-Handler pathway in their honor. The non-redox roles of NAD, P, are a recent discovery. The first of these functions to be identified was the use of NAD+ as the ADP ribose donor in ADP ribosylation reactions, observed in the early 1960s. Later studies in the 1980s and 1990s revealed the activities of NAD+ and NADP+ metabolites in cell signaling such as the action of cyclic ADP ribose, which was discovered in 1987. The metabolism of NAD+ has remained an area of intense research into the 21st century, with interest being heightened after the discovery of the NAD+- dependent protein deacetylases called sirtuins in 2000, by Shinichi Oomai and co-workers at the Massachusetts Institute of Technology. See also, enzyme catalysis, list of oxidoreductases, references. Further reading, Function Nelson-Dale. Cox M. Leninger Principles of Biochemistry W. H. Freeman. ISBN A0-7167-4339-6 Bugti Introduction to Enzyme and Coenzyme Chemistry. Blackwell Publishing Limited. ISBN A1-4051-1452-5 Lee H. C. Cyclic ADP ribose and NAADP, Structure, Metabolism and Functions. Kluwer Academic Publishers. ISBN A1-4020-7281-3 Levine O. S., Shahota, Schwartz B., Wenger J. D., Elliott J. Centers for Disease Control. Generic Protocol for Population-Based Surveillance of Haemophilus influenzae Type B. World Health Organization. PA 13. Wagen/95.05 History, Cornish Bowden, Athol. New beer in old bottle. Duarte Buchner and the Growth of Biochemical Knowledge Valencia, Universitat de Valencia. ISBN A84-370-3328-4 A History of Early Enzymology. Williams, Henry Smith, 1863–1943. Modern Development of the Chemical and Biological Sciences. A History of Science, in 5 volumes 4. New York, Harper and Brothers A textbook from the 19th century. External links: NAD bound to proteins in the PDB, NAD animation, I-squared nicotinamide adenine dinucleotide and NADH chemical data sheet from Sigma Aldrich, NAD+, NADH and NAD synthesis pathway at the Metasec database, list of oxidoreductases at the Swiss PROT database.